typical retrovirus RNA genome looks like this. It has two ends, called long terminal repeats, LTR, and only 3 genes: GAG, POL, and ENV. The LTRs are sequences that help the virus integrate into the host genome. Once integrated, the virus uses a host replication machinery to produce RNA containing the LTRs and genes between them and the other viral proteins. POL codes for the reverse transcriptase. GAG encodes the capsid and other core proteins, while ENV produces components of the outer viral envelope. The proteins are then assembled into a viral particle with the viral RNA inside. Once it leaves the cell, the virus is capable of infecting other cells. Tumor viruses, like RSV, have picked up an additional gene called an oncogene. SRC contributes to malignancy. Crick's central dogma, where information flows from DNA to RNA to protein, does not apply to viruses like RSV. A reverse flow of information from RNA to DNA happens first, and these viruses thus became known as retroviruses. The discovery of reverse transcriptase helped to clarify the life cycle of retroviruses, which consist of an outer envelope, an inner capsid, and a central core containing the RNA template and the reverse transcriptase enzyme. When the virus infects a cell, the viral envelope fuses with the host cell membrane and the capsid disassembles, releasing the RNA and reverse transcriptase into the cytoplasm of the host cell. The reverse transcriptase then synthesizes a complementary DNA strand using the viral RNA as a template. Then the enzyme degrades the RNA and synthesizes a second DNA strand. The DNA makes its way into the nucleus and integrates into a chromosome of the host cell. Retrovirus were discovered by David Baltimore and Howard Temin while studying of Rochus sarcoma virus, RSV, that cause tumors. RSV is named after Peyton Rouse. In 1911, he found that this virus causes tumors in chickens. Rouse passed an extract from tumor cells over filters that excluded any material larger than a virus. This filtrate produced tumors when injected into healthy chickens. The implications were that viruses cause tumors, but no one knew how the viruses worked to cause cancer. In the 1970s, David Baltimore and Howard Temin proposed that, during infection, tumor viruses integrate their DNA into the host cell DNA. However, this would prove a problem for RSV, which stores its genetic information as RNA, not DNA. They became interested in how RSV might use its RNA as a template to make DNA. To figure this all out, they stripped the virus of its outer coat and used the inner core in a cell-free system. They added all the nucleotides. Thymine was the radioactive tracer. The viral components in this cell-free system produced radioactive DNA. They added RNase, as they knew the radioactive product was DNA because it was not affected by RNase. But the product was destroyed by the enzyme DNase. They incubated the viral cores with trypsin, an enzyme that digests protein, then added nucleotides to the trypsin-digested viral cores. They did not get any DNA product. Similarly, if they added RNase at the beginning of the reaction, the added nucleotides, there was also no DNA product. They concluded that RSV has a special enzyme that uses RNA as a template to make DNA. It was initially named the enzyme RNA-dependent DNA polymerase, but it later became known as reverse transcriptase. David Baltimore showed that retroviruses have a gene for reverse transcriptase called POL and also carry the reverse transcriptase itself into cells. In his experiment, Baltimore showed that adding trypsin to the viral particles destroys the reverse transcriptase and no DNA is made. The viral particle didn't make any reverse transcriptase as there was no tRNAs, ribosomes, and no amino acids. Retroviruses can be used as vectors to deliver new genes to animal cells. First, scientists insert DNA with the GAG, POL, and ENV genes into the DNA of cells in culture. These cells are called producer cells because they produce the viral proteins, GAG, POL, and ENV. However, they do not produce viruses because there are no LTRs. LTRs have several functions. They not only help integrate the viral genome into the DNA of an infected cell, they also indicate where RNA transcription begins and ends. Without LTRs, no viral RNA is produced. Without RNA, there is no virus. The next step in turning a retrovirus into a vector with a new gene. To transform the producer cells with a plasmid that contains the new gene. Once transformed with plasmid, these cells produce modified virus. LTRs bracket the sequences that will be made into viral RNA. The producer cell DNA lacks LTRs, so the GAG, POL, and ENV genes are not transcribed into viral RNA. LTRs on the plasmid DNA initiate RNA transcription of the new gene and LTRs. Suppose there is a genetic disorder where white blood cells do not produce a needed enzyme. Retrovirus as a vector to deliver a new gene to the patient. Inserting the gene into bone marrow stem cells can make the gene transfer permanent. The reintroduced stem cells carry the gene and will pass it on to new cells they create, including future stem and bone marrow cells. Because the stem cells only make white blood cells. For technical reasons, scientists have not successfully transformed bone marrow stem cells.